guys can have a more independent work time today. So let's do this thing.
Excuse me?
Yeah, Sarah, what's up?
Is it okay if I leave the class a little bit earlier today? Because I board a flight in like half an hour, like so. So like towards the end of class.
Like what? Where are you going?
I'm coming back from Atlanta.
Oh, is it nice in Atlanta?
Yes, it is, but it's really, really cold.
Oh, okay. Yes, that is fine, sweetie.
Okay.
Yeah, everything will be posted on Google Classroom. So yeah, just catch up from there.
Okay, I will. Thank you.
Mm-hmm.
I can see you now, Morgan.
Okay. So, yeah, I can see you too, Morgan. Here we go. So we are at, it is not February 2nd. What am I thinking? It is February 3rd. And it is Wednesday. We are going to be kicking off with the first official activity in your notebook. So if you guys would be ever so kind to head on over to Google Classroom, and you're going to open up your copy of the protein synthesis digital notebook and get yourself on over to slide number 4 for me. For this unit, we're going to start off kind of like where we were at yesterday with the our funny little Kahoot. We were looking at DNA. We should have gotten the gist that it's DNA is the basis for our genetic information. Lots of crazy things with DNA. And we're going to dive in today a little bit more just about the basic structure and organization of that awesome molecule. There's this overarching theme in science, and it doesn't matter what area of science you're talking about. It's the idea that structure and function go together. The way something is built and put together dictates what it's able to do. So structure meets function, and they're like married together. Okay. That is completely true for DNA. Okay. So the way the DNA is put together and the way that it's built actually allows it to function as your genetic material. So we're going to look a little bit about, we're going to dive deeper with that today. So the big things, your big takeaways and goals for today and into tomorrow are these 3 things. 1, you'll be able to define DNA. So be able to tell me what it is, and you'll be able to define the term nucleotide. Okay. Second thing, you'll be able to model the structure of DNA. So you'll be able to identify its parts and kind of explain the bigger like pieces of a DNA molecule. And 3, you'll be able to explain like what are the reasons behind those big structural pieces? Like what is the significance of the middle? What is the significance of the outside portions of it? Okay, so that's our big kind of to do for today and into tomorrow. Cool. If everything else kind of you're like, I don't understand anything else, but you get that, you're on track. Now, the way that we're going to start out with this though, is that you guys, we're going to play with, we're going to do a puzzle. Now, I'm kind of geeky and I love puzzles. Like that's my nothing box. I just do puzzles and like brain teasers and stuff. So we're going to do a puzzle together. In Google Classroom, under the lesson 1, you're going to have a copy of the building DNA. It's a Google slide. So everyone's going to open up their own individual copy. You guys are I'm going to give you 5 minutes to put your own puzzle together. Okay, using a set of puzzle pieces. Now, if I go over to that, okay, so if we go to lesson 1, it's going to be right here where it's going to say building DNA. So it's going to make a copy for you guys. On the perimeters here, those are your puzzle pieces. Your goal is to get all of those puzzle pieces to fit inside your playing field right here. You guys figure that out. So however you can figure out how to get it in there, that's how you get it in there. But I've set the first 2 in there for you so you can use that as your template. Okay, so you got 5 minutes to arrange these guys in there. Notice how they line up on the side. So this is, you've got to fit them all inside this middle region. They can't hang out on the edges. You got to fit them in the middle. And they've got to sit tight up against each other. Okay, so that's your challenge. Now, a couple of you already did it from yesterday, and so I have that. If I left a comment on yours, you can open it back up. And if I said, hey, let's try this one again, go ahead and you can redo it. If I didn't, or if I didn't reopen it for you, I will right now. So hang tight. I think Jason, I might have to if you didn't submit it, or if you did submit it, I'll return it to you so you can do that. Okay. So go in there. Let me get your screen up for you guys. You're going to have 5 minutes. I'll set a little timer for you. So you guys kind of are going to be able to follow along. Once you guys are done, I'm going to share a couple of your guys' work. So once you're done, I want you guys to hit submit. Okay. And then that way we're going to kind of compare and contrast what you guys come up with. And then we'll use that information to go through the questions on slide 5. I should say, if you get done early in your notebook, okay, on slide 5 is where we're at. So if you get done early, we're looking right here. We're going to do this stuff right here. Okay. So you're going to be filling out and kind of like going through the different characteristics. So what are some things that you notice about the pattern? We will do this together, but if you get done early, you can start looking at those questions, but we will answer them as a group. Okay. So if you get done early, you can do a little bit of that. All right. So you got 5 minutes to see if you guys can put that puzzle together. All right. Ready? Go. Go for it. And if you see me popping in on your notebooks, I can get in there and look at yours. So don't be alarmed. It's like me monitoring what you're doing. Walking around the class right now. All right, you guys are at just under a minute, just under a minute. All right, guys. Okay, so let's take a look at some of your guys' creations here. Okay, so into your notebook, let's figure out. So I want you guys, I'm going to open up some of them. Okay, and I'm going to have you, and there's random order, whoever had turned theirs in. So if I pick yours, like it's just because you had it turned in. And I will tell you, they're absolutely everybody who's turned it in is 100% correct. Okay, so I want you to compare yours to the person that I am sharing out. Okay, so let's go in here. Let me open up your guys' work. Okay, so let's go first. We'll go to Collins and see who's first on that one. All right. A couple of things. So the first question on our table is asking us to look at some patterns that have to do with the pairing. So when you guys are looking at the pieces themselves, those pieces are little puzzle pieces. Those guys are called nucleotides. And if you remember back to, oh my gosh, what would it be? First quarter, your nucleotides are the building blocks of your nucleic acids, which is what a DNA molecule is. These are the building blocks for our genetic materials. So your nucleic acids, we have 4 of them. Okay, we have A's, T's, C's, and G's. They actually do have proper names, which we will get into, but we usually refer to them by their shorthand abbreviations, so just by their first letters. Now, all living organisms have DNA. That's one of our connecting characteristics for everything. So it doesn't matter if we're talking about bacteria, viruses, trees, cats, dogs, whatever, everything. We are all made up of the same 4 building blocks: A's, T's, C's, and G's. Okay, just these 4. Now, what do we notice about their pairing? What goes together? So it doesn't matter, like if you're looking at this one, or let's say Eliana's, okay, when hers loads, what do we notice about what actually fits together? You guys can either unmute yourself or drop your comments in the chat. So what do we notice about what letters actually fit together? All right, so Jillian, so A's go with T's, okay? Marcella, G with C, A with T's. Definitely. Notice that we could click on anything. And notice how this one right here, so Ellie's diagram looks absolutely 100% different than Colin's, which probably looks 100% different than the one that you created. But like I said, all of them are correct. But the thing that they all have in common is that all the A's and T's are linked together and all of the G's and C's are linked together. That is a constant, something that always, it's something set in stone. A molecules will always be paired with T molecules. G molecules, G building blocks will always be paired with C building blocks. Always, 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 always. We call that our base pairing rules. It's a rule, okay? So in our notebook, in that first one, we're gonna kind of put that rule in there. We're going to call it the base pairing rule or rules. Now I like to just make it bold. Base pairing rules. So A's always go with T, and G always go with C's. Okay? Now, that is something that's always consistent. Questions on that part? And these are vice versa, so we could always say A goes with T and T always goes with A, so you read it back and forth. Now, the next thing, what do we notice about the sizing? Okay, the sizes of the molecules. We'll go to another person's here. So what do we notice about the sizing of those, the building block pieces, your puzzle pieces? Yeah, Ravens. Some are thin and some are thick. So you're talking like the depth. So some are long and some are short. Yeah, Morgan. Some are shorter, some are longer. Notice how which ones are going together. Okay. The only way that you were able to make this fit. So if I started messing with Ashlyn's here, you would never see an A and a G together. Okay. And we would never see a T and a C together for a couple of reasons. One, aside from the fact that their little middle regions don't fit together, which obviously your DNA doesn't fit nicely and have little like jigsaw puzzle designs in it. But notice how it doesn't keep a consistent shape. We don't stay within this boundary. For your DNA to stay its proper shape, it has to stay the same width the entire length of it. And we're talking, remember, it's 2 meters long. So the only way that it stays like that is if it is bonded with something and that pattern stays the same. So a long one with the short one, long one with the short one. Okay, so 2 short ones can never go together because you would get these dips in its outer edges. 2 long ones would never go together because then it would push too far wide. All right. So when you're looking at the pattern there, okay, it is always 1 long one with 1 short one. So the ones that are your fat ones are always going to be your A's and your G's. Okay, your skinny ones, your shorty ones are always going to be your C's and your T's. Okay, so the short, we'll call them nucleotides, are T's and C's. The A's and the G's. I'll look that up so we can see that better. Oopsie. There we go. Okay. Which makes sense. So, because if we go back to our base pairing rules, a T and an A will always go together. So we always end up having 1 short and 1 long. Now, the really cool thing when we tie these 2 things together, now, if we notice this shape that if you ever look at any diagram of a DNA molecule, it creates what we call a double helix. So it looks like a spiral ladder. It makes this ribbon effect. Now, the reason why it does that, almost think like too, if you took a slinky and you stretched it out and it makes that little like the slinky thing, right? It makes a little spiral. Now, DNA does that because it's hooked with 1 short and 1 long. It kind of makes it like slant a little bit. So, but when it's hooked like that and it's slanted, it causes it to turn on itself. And that's why the DNA molecule gets that spiraling shape, is because we've got 1 long molecule hooked with 1 short molecule, and it causes it to kind of be off kilter, and it causes it to slip and go whoop, and spin downwards. Okay. These things right here in this box are things that are constant and consistent. They're never changing. No matter what sort of living molecule or organism you're talking about, these are things that you'll always find, okay? Now, the last one has to do with the direction of the molecule, okay? And the way that your nucleotides or your puzzle pieces faced. So what do we notice about the direction of those puzzle pieces? Let's see if I go back. Let's see. I'll go in here. Okay. So what do we notice about this direction and the way that those puzzle pieces are arranged and the way that they're facing? I guess what does this column all have in common in the way that they're facing compared to this column? Yeah, Ricky, yeah, Morgan. These ones are all upright. Okay, they're like, they look like they're the normal ones. They're all upright facing, they're like straight up. Compared to their other side, okay? Notice how this side, they're all upside down, all of them, okay? That is also on purpose. So this has a special name. So this is called, they're parallel, okay? So just like in math, you have two parallel lines. So they're equal distance apart, meaning they're never gonna touch. So they're never gonna overlap each other. They're never gonna run in. They're always going to be the same distance apart from each other. However, they're running in opposite directions. In science, when we say something is opposite, we usually use the prefix anti. So we would call these things anti-parallel. So DNA is anti-parallel, meaning that one strand of your DNA runs in this direction, so from up to down, and the opposite strand, or its partner strand, runs from bottom to top. So they run in opposite directions, almost like if you had a zipper, it's like zip, you're zipping them in opposite directions. Like if you guys ever had that jacket that has a double zipper, like you can go up or you can go down, it's like that. It's kind of like, but it zips itself in opposite directions, which is really cool when we have to open, when your body has to open and take a peek at its DNA to get some codes out of it. Okay, and then that's what we're going to look at tomorrow and how that works. Okay, so we go back to your notebook. We would say one strand is, we'll call it, you guys use the word upright. The opposite, or the other strand, is upside down. This is called anti-parallel. Again, oops, I spelled that wrong. This just means they're parallel, but running in the opposite direction. Okay, so how are you guys feeling on those rules? Now, when I say rules, I mean legit rules. No matter what organism's DNA that you're looking at, you're always going to find that those things are present. Always will see A's with T's. Your T's and your A's are always going to be long, and your C's and your G's are always going to be short, always. So if I gave you any random piece of DNA, you could use those rules to help you label and figure out what was what. Okay? So really quickly, let's go ahead, because this is really what we're going to start looking at in the next piece, is this. Your DNA, the code, that all the instructions that make you, have to do with the sequence of those letters. Okay? Now, if we had to look at the sequence, though, we know if we had one strand, we can have the opposite strand. So it's like if you had one side of your puzzle, you could build the other side of your puzzle. So using the base pair rules, okay, and knowing that A's always go with T's and C's always go with G's, what would this partner strand be? Okay, so kind of help you get started out. So A would always go with T, R. C would always go with G, G always with C, T's always with A, B's always with G, A is always with T, with A, B, C, A, B, C, D, A, A, B, A, T, C. Okay. Now, this is the really cool thing with your DNA. When you have one strand, we can always determine the opposite, or what we call complementary strand, because they always go together. And I apologize if that is small. I can try to make it a little bit bigger. Okay. Now, the other thing, while I kept saying we've looked at these things where we're like, okay, with DNA, there are a set of rules that have to do with its structure, right? So far we've got these rules. Okay, we know that A's will always go with T's. There's always a big and a small together. They're always going to run in opposite directions. Now, that's great, but where does the diversity come from? Yeah, you can use ours, or use mine, Sabita. Okay, so where's our diversity? Why do we look so different than the trees outside? Why do we look different than our dogs and our cats? Okay. And I said before that every single person who turned in theirs was absolutely correct. And it has to do with the way that you put your puzzle pieces together and the way that you stacked them. This is the missing piece. This is like where the magic happens. The order in which you put your letters, your nucleotides, your bases, the way that you stacked them. This is where your unique qualities come from. This is your code. So every organism, okay, has a different pattern in the way that their nucleotides are stacked and put together. So the order that these guys are in is different. Okay, so there's lots of similarities, there's lots of difference. So remember in our Kahoot that we said that, okay, 99.9% of our DNA is the same. So all of ours would be the same except for that 1%. So we could say, okay, Ashlyn's 0.1% looked like this. We would say that Jillian's 0.1% looked like that. And this made all the difference in the world. And Neza's 0.1% was this repeating pattern. Okay, completely different. There's some similarities maybe, but it could be completely different. Okay, and they followed all of the rules. They had their A's and T's together, C's and G's were together. They stayed within their boundaries. They created their appropriate shape. Notice how Colin's was completely different. He had his stacked and repeating orders. Absolutely acceptable. This was his differences. So this right here, the order in which these are arranged is our 0.1%. This is where all that magic happens. And it's right here, there are the instructions for making you. Okay. More specifically, this is the instruction manual for making the proteins that make you. Okay. Questions on that part before I set you free? So our whole unit is going to be this link between, okay, we know we have this instruction manual for you, and it's this sequence of these crazy letters, this hidden code that lies in all of your cells, almost all of them. So how do we go from these crazy molecules of C's and G's and T's and A's to you? Go from proteins like DNA to proteins to you. So how does that work? That's where we're going to go. Okay. So in your notebook, I'm going to give you some independent time that you guys are going to be able to finish that up. So for the remainder of the class, I'll have you guys finish up slide 5 if you still need to. Okay. And I'll leave this up if you guys have to pop back into class. You guys can copy that down. But you guys are going to be responsible on your own for completing slide number 6. If you did this yesterday, then you are done. You're ahead of the game. If you did not do this yesterday, which you shouldn't have, because I didn't assign it to you, then that's what you're working on right now. You guys are going to be watching a short video. It's literally 4 minutes, so it's not too crazy. And it's posted in Google Classroom under the Engage lesson 1. And it says DNA structure. As you watch, you can either have it like side by side, like split screen, or you can pop back and forth, or whatever strategy works best for you. But you're going to fill in these boxes as you watch the video. So it goes a little bit more in depth about what we just covered, giving proper names and going a little bit more, naming the parts of your building blocks. So the puzzle pieces, your nucleotides. This right here is more of a proper, so a step up from the puzzle that we just did. So these were your pieces, but each piece is actually made up of a 3-part molecule. Okay, so after you watch the video, you're going to then drag these little labels into their correct spots. Okay, and then once you are done with that, you are done for the day. Okay, and then tomorrow we'll go through it and we'll start to add a little bit more to this page about what's the significance of this region on the outer edges, what's the significance of this region right here, and about its structure and its function. Okay. All right. Questions? All right. Remember, you guys are free to stay here. At this point, you guys can leave your cameras on or turn them off, whatever you feel most comfortable with. You can stay on and work. I can put you into breakout rooms if you want to work with your friends. That's totally fine. You can leave and go work on this on your own. The expectation is that you come to class tomorrow and slide 6 is completed and you've watched the video. But once these items are completed, you are done for me for today. I will be on in the meet until 1:45. So if at any point you have questions, feel free to jump back in, get your questions answered. Yeah, but other than that, that is all I have for you today on our in-class stuff. So you are free to go and finish the rest of it on your own. Other than that, take care. Have an amazing day. If you guys do have questions, stay on and I can help you get those things answered. Other than that, bye, awesome people. And yes, Savita, I will jump back to slide number 5 so you guys can finish copying that down. I realized I was probably going a little bit fast. Let me make that big for you so you can see. There you go. And the little bottom part will go away here in a second. There you go. And bye, everybody, as you're leaving.
Bye.
Bye.
Bye, Miss Cannon.
Bye, Ineza.
Bye, Miss Dijon.
Bye, Ian.